Yeah. Okay, first of all, I would like to thank Jim for this very, very nice words. And also like to thank the organizer committee for inviting me to come here and talk about swine, or fiber. Fiber in respect to the functional and also nutritional properties in swine diets. This is an area which I've been working with for many years. I've been into various aspects of the fibers, first from a very analytical perspectives, then from doing experiments on how different fibers and carbohydrate components, they have been digested and absorbed from the various parts of the gastrointestinal tract. Then I have had some projects together with other groups where we looked on interactions of the carbohydrates related to the establishment of parasites and also gut health. And here more recently, because of the funding situation in Denmark, I've had strong links with Aarhus University Hospitals after we became first a faculty underneath Aarhus University and now a member of Science and Technology with those in human nutrition. But the points which I'm going to address here today, that is some words about fiber terminology, chemical structure and physiochemical properties, nutritional properties of fiber, and also how it impacts digestion, absorption, and utilization of carbohydrates-derived nutrients. What I've chosen to do, that is to collect a number of data which we over the years has generated, first of all, on carbohydrates and lignin in different feedstuff materials. This is works that is published and you can find them in the literature. A number of data from ileal cannulated pigs where we link carbohydrate composition to also how we see the data on more quantitative aspects from ileal cannulated pigs. This is data which I've collected as part of our own results, but it's also part of results that has been published, but basically they are based on publication where they have been using approximately the same analytical techniques as I'm going to demonstrate later on. So you will not find any data on NDFs and so on. And then finally also data from catheterized pigs, which is based on results we have been doing where we have been looking on the impact on how different carbohydrates they may influence the assimilation of carbohydrate products, that means uptake of starch, and some of the fermentation products as lactic acid and short-chain fatty acids. And finally, I also have some links between carbohydrates in feedstuffs and also data from feedstuff evaluation, which is some older works that has been performed at our institute. And I should though mention that this link is not in your proceedings because this is not published yet, so you have to wait for that. Some general introductory remarks. You see dietary fiber present in almost all plant materials. They also represent the part of the diet that cannot be digested by the endogenous enzymes, but they can to a potential at least be fermented by the microflora. And because of the lower utilization of energy that derives from microbial degradation products, that will also have an influence on the energy value of the feedstuff. There is also so that there is a number of physiochemical properties that's associated to the dietary fiber fractions, that's the swelling, water binding capacity, and also viscosity. And there is also so that there are age differences in how well the animals can cope with high fiber diets, meaning that older animals, they have a larger capacity to ferment fibrous component, primarily because you have a longer retention time within the gastrointestinal tract, and maybe also to some extent because you have a slightly different microbial flora of older animals. The last one I'm not going to address that much today. If you look into the carbohydrate composition of the diets and also look on what is the fiber levels in diets for piglets, in diets for growing finishing pigs, and for sows, you'll see that it goes from between 8 up to 14%, and now when I'm talking fiber here, I'll come back to that a little later on, it is actually diet or the non-starch polysaccharides and lignin content of it. It's slightly lower when you come to growing pigs, and for sows it's also a little bit higher again, and you can have a much wider range on the amount of fiber you can find in the diets. I would like to state that the fibers or the diet is not a well-defined chemical entity, but it is a term which in animal nutrition which traditional has been defined by the methods applied for the analysis. And there is a point that's very important, because depending on the methodology you are using, you will get many different values in terms of fibers. And you'll see a lot of different values in the literature, and also different values for the same type of feedstuff materials. Those which traditional has been used, that is the crude fiber methods, that's the very old methods. There is the detergent methods developed by Van Soest and co-workers, including neutral and acids detergent fibers. There is the enzymatic gravimetric AOSC methods, and then there is the enzymatic chemical gravimetric methods. This is basically some of the methods we have had and which are around at the moment. And if you come to the definition, as I mentioned before, it's not a term that really has been defined in the animal nutritional literature. But here in the human nutrition, they have finally, after 30 years of discussion, come to an agreement on the definition of dietary fiber. And you can read that this is the Codex Alimentarius and also the European Commission, which states that dietary fiber, that is carbohydrate polymers with 3 and more monomeric units, which are neither digested nor absorbed in the human small intestine, and belongs to the following categories, and so on. Here there is actually also lignin included. I should say I'm just briefly attaching this one, whereas for most part I'm working or I'm presenting data that's more related to non-starch polysaccharides and lignin. Because what does it matter if you change the definition from what we more classically consider as dietary fiber towards the new definition? Well, first of all, going back to the presentation of Dr. Steins, he talked about the raffinose oligosaccharides. In the new definition, raffinose oligosaccharides, they are part of the dietary fiber definition. So they include the families of raffinose oligosaccharides. It includes fructooligosaccharides and so on. But for instance, the components as maltodextrin, they will not be part of, because that will not be resistant. And then it also includes resistant starch and those parts here which we traditionally will consider as part of the fiber matrix. This, as I mentioned before, this is what you get if you use different analytical techniques. You'll see that you can have from low values here for the crude fiber methods, acid detergent fibers, neutral detergent fiber, including more of what is part of the cell wall materials. And then you can also divide them into insoluble and soluble, and you can use other techniques to specify it even more. I should say the methodology we have been working with in many years, they are related to this technique over here. I will not say that the perfect method to do. It is a tedious procedure, but it gives you good information, particularly for experimental purpose, on how different carbohydrates they behave in the gastrointestinal tract, and also in particular when you should try to interpret results from nutritional studies. How will it look if you take on some of the different methods? Well, if this is the new definition according to alimentaric, the new human definition, you'll see here, if you take the old one for barley, this represents there is 5% less if you use the non-starch polysaccharide and Claassen lignin method. Neutral detergent method is lose around 30% less. Acid detergent fiber 27 or 63% less, and what is that? That's 7-8% less. This is for barley. But if we come to a soybean meal where we have much higher levels of oligosaccharides, you'll see that the difference become even more pronounced. Chemistry and methods. Well, first of all, what I'm going to talk about now in the remaining part, that is primarily the fiber components that is measured as non-starch polysaccharides in lignin. And where do they come from? And where are they located? Well, the fiber components, they are present in the cell walls, primarily. I should though say that, for instance, if you analyze plant materials as lupin, you will also have a lot of non-starch polysaccharides present outside the cell walls. But that's some of the few exceptions. But basically, they are present in the cell walls. And what do they consist of? Well, they consist of a number of different polysaccharides which are linked to each other and which makes up the cell wall structure here. And what you can see here, that is that in the cell wall structure, they also surround cell content. And that is very important when we should try to understand how can the fiber components potentially influence also other components of the diet. The cell walls, we do not have any enzymes present in the small intestine that can break the bondings in the cell walls. And as long as the cell wall, they stay intact, principally, you cannot digest the content. When we grind the materials, we will have some of the cell walls broken, and we will also, because some of the cell walls, they are more hydrophobic in the nature, they will start to dissolve, meaning that there will be possibilities for the endogenous enzymes to penetrate the cells into the cell content. So they consist of a number of different polysaccharides here. What we have been working with for many years, that is an enzymatic chemical gravimetric procedure. It is basically based on some of the ideas that were laid down in procedures of Hans Engelens from the UK and also from Per Åhman and Olof Theander from Uppsala. But given this classical, I would have drawn the line here, say this is the fiber components, and that is also what I'm going to do today. But don't forget, these parts here with the new definition, they are also part, at least on fibers in humans. But they are based on, we have enzymatic methods by which we can measure, or HPLC methods by which we can measure the sugars. We can also do that for the oligosaccharides. Starch divided into starch, total starch and resistant starch, we can do by enzymatic methods. Fructans, we have either HPLC or enzymatic methods, and then we have the whole complex here of plant cell walls where we can use enzymatic, chemical and gravimetric methods to quantify them. What you can see here, that is, by using that, we divide them into cellulose and non-cellulosic polysaccharides that can be soluble and also insoluble. And these are also what in the literature also are named as hemicelluloses. It's just different names. And then we have the lignin, which, although this is not a carbohydrate but a phenolic polymer, it is very important because the lignin in the lignified tissue, they are closely linked to the polysaccharides. And the more lignified the tissue are, the more difficult it will be to degrade. Not only for the microflora, but they will also have an impact on the physiochemical properties, because if it's so that we have a high content of lignin, we have a lot of cross bindings, that means that the cell walls are less elastic in the way they are behaving. Functional properties, I've already been around them. They protect the cell contents against digestion, as I mentioned, present here in the cell walls. When they are exposed to water, they hydrate. During that process, they swell, and some of the fiber components will leak out into the liquid phase, which is what is giving us the viscosity of the liquid digester. And the fact that cell walls can protect the content of it is not a new idea, because here's Saunders, who worked at USDA in Western, somewhere in Berkeley. He already in '69 was looking on cell structures and how that influences the digestibility of nutrients in chickens and so on. Just a little bit more about the hydration properties. Well, first of all, it is when water come in, and you should imagine that how a cell wall can swell in a three-dimensional space. When water comes into this matrix in here, they start to loosen up some of the cell structures, and those of the cell structures that can leak out into the solution, they are what giving the viscosity, whereas the remaining part will be trapped within the cell wall matrix. Here's trying to illustrate how it looks when you take a certain amount of oat hulls, high in insoluble fibers and high in lignin, and you do the same with sugar beet pulp, which has the same, a little higher content of fibers, but which are non-lignified tissues, and you can see some of the differences in how much of the volume of the water the two fibers they can hold, or at least they can surround. Generally speaking, it's so that in most of the cotyledon, potato pulp, pea hulls, and sugar beet pulps will generally have a higher water binding capacity and also a higher swelling capacity than fibers from cereals. You'll see some of the differences here, although this is also quite fibrous material. The reason, as I explained before, in the lignified tissues is so that the cell walls that's in there, they are restricted in their expandability. What does it matter? What does it mean? Well, if we look on the effects of fibers on the total volume flow of digester through the gastrointestinal tract, and here is a collection of data from different experiments feeding at low levels of dietary fiber, intermediate level of dietary fiber, and a high level of dietary fiber. And what you can see here when you're looking on the flow of digester, that it goes up as the fiber levels of the diet goes up. It goes up, and what's happening here, that is that the starch and sugar that's present in the diet, they are depleting during passage of the small intestine, and what is up or concentrated in ileal digester, that is the fiber components. Nutritional properties. Well, first of all, if we look into what is the potential properties of fibers on the digestion and absorption of nutrient in the gastrointestinal tract. Well, because of the water holding capacity of the fiber components, it may have an impact on the gastric emptying. Fibrous products or high fiber diets will usually stay in the stomach a little longer. It decreases the rate and extent of nutrient absorption from the small intestine. And then, as I showed before, it increases the flow of digester to the large intestine. And because the fibers represent the part of the diet that's not digested by the endogenous enzymes here in the small intestine, it increases the fermentation in the large intestine. There is simply more nutrients coming down to the large intestine for fermentation, and it also reduces the total tract digestibility and the transit time and so on. I'm going to present some data here which are from 21 published work, and then also one unpublished works with ileal cannulated pigs. As I mentioned before, these data here have been selected based on the fact that they have been performing digestibility with ileal cannulated pigs, mostly with ordinary cheek cannula placed in the end of the small intestine for collection of digester. In most cases, they have been using a marker to look on as a marker for the indigestibility. And then they have been analyzed with methodologies that allows us to get information about non-starch polysaccharides and also lignin content. In some cases, there is also further information concerning the other components as starch. That's actually the case in most of the studies. And then there is also some studies that include data on some of the oligosaccharides, but that is fewer. Well, first of all, we have the digestion here in the small intestine. We have the alpha amylases, and we have the enzymes on the epithelial cell border. The products that come in, that is glucose, whereas when you have the fermentation going on in the large intestine primarily, but there is also some fermentation going on in the stomach and the small intestine, the products that is short-chain fatty acids, lactic acids, and gases. This is how it looks when we come across all the different data. And what you can see that is that sugars and digestible oligosaccharides, they are almost completely gone at the end of the small intestine from those where we have this data. Non-digestible oligosaccharides, a split that says approximately 40% of non-digestible oligosaccharides, they will be degraded during passage of the small intestine. The remaining parts go to the large intestine. For starch, it's so that by far the highest content, they are digested in the small intestine. But even for non-starch polysaccharide, you will see that we have a loss during passage of the small intestine of approximately 22%. That is due to the microflora that's colonizing the upper part of the gastrointestinal tract. What I then have tried to do, that is to relate the intake of fibers to the flow of nutrients at the end of the small intestine. And what you can see here, you have the organic matter on average, and they have been fixed with an intake of 2 kilogram per day. You'll see that what reaches the large intestine, on average, organic matter 475 grams. But you will also see here, if you look on the effect of dietary fiber, and here, as you said, there says non-starch polysaccharides and lignin, you'll see they are closely related to that. Concerning the carbohydrates, you can also see there is a strong link between the intake of dietary fiber and non-starch polysaccharides. That's also to be expected. But you can also see here a link relative to that of protein, whereas fat is purely related, and the same is actually the case with starch. So, and that is when you're looking on how is the digestibility in percentage of organic matter and nitrogen, and this is apparent digestibility. You'll see the significant negative correlation. And why is that the case? We find this here. Well, this is not surprising, because this is actually just because we have more non-starch polysaccharides, where we may want a little bit more. Why is that the case? We find over here in particular some of this is very, very low. Well, first of all, because there is 2 effects of the fibers of it. First of all, it encapsulates, or at least potentially can encapsulate the nutrients. And what I've shown up here, that is from an experiment that was performed, and where we were feeding the animals oat bran. Oat bran, that is the oatest part of oats. And we then collected the ileal digester. And what you can see here, that is how it looks in the feet. I said the red one here, that is the beta-glucans that is present, and then we also have the same digester collected at the end of the small intestine. And what you can see here, that is that they are actually heavily loaded here with protein and fat in the cell walls from the aileron part of the grain. Another factor which also plays a role, that is the viscosity that can be generated with some types of products or feeds, as for instance the case here with rye, which can give rise to relatively high viscosity in the small intestine. They will influence the endogenous secretion of nitrogen to the small intestine. As I mentioned, starch no relation or a small relation to the total intake of dietary fiber, at least in these studies here or in the studies I've been over. What really matters for the digestibility of starch in the small intestine, that is the particle size, and then also the digestibility of starch from legume, that is substantially lower than what you see general for cereals. One cereal or one type of cereals, though, which can result in lower starch digestibility, that can be starch that is high amylose types of starches, where you also can have lower levels of starch digestibility. Moving to the large intestine, it's a moist environment, high number of microorganisms, long retention time, anaerobic conditions, and which means that there is a long time for the microflora, which has a much higher bunch of different carbohydrate degrading enzymes which can attach to the cell wall materials, and then can use the enzymes to break down the different components in the fibrous matrix. And when I'm looking on all the different data, this is approximately what I typically find. Sugar, starch, and the most of the non-starch polysaccharides, they will be fermented. All soluble non-starch polysaccharides, they will be fermented in the cecum, proximal part of colon, and that means that they are fermented, they are broken down to short-chain fatty acids, which are absorbed to the body. In the order of 95% of what is produced here, they will be absorbed to the body. The other part of the lignified part or those which are present in lignified types of materials, they are far more resistant to degradation, primarily because you have the lignin that cross-linked to the different cell wall structures. And although you can have a long retention time in the gastrointestinal tract, the major part of that will actually be the ones you find in the fecal materials. And that is also what particularly gives rise to what they in human nutrition will talk about the fecal bulking and also the bulking within the gastrointestinal tract. Well, this is basically what we have been seeing when we are going over many of the different experiments. There is a gradient here on how they are metabolized. It's so that, as I mentioned before, those which are relatively easy for the microflora to degrade, they are degraded in the cecum and proximal colon. There is other parts that is more difficult to degrade more slowly, and some of them will be present in fecal materials. And this is approximately what is happening when we have high lignification of the materials. Well, the thing is that in spite of the fact you have a lot of different microbial enzymes that's able to cleave the bondings in the plant carbohydrates, as the degradation progresses, you'll come to a state where most of the cell wall matrix, they will be covered by lignified materials, and which just will not make it possible for even the microflora to degrade these type of substances. Generally speaking, it's so that sows will have a higher capacity to degrade the cell wall materials than is the case for growing finishing pigs and also piglets. I have not figured out completely why that's the case, because when I try to track, for instance, degradation along the length of the large intestine, it's actually so that there seems to be plenty of time for it to also handle some of the same carbohydrates as the sow can do. Maybe the key point, that is, as an animal mature, get older, they got a different population of microorganisms. Again, if we try to look on relationship between the fibers, you will also see that they goes down here. And if I just am moving on to the next part here, which is a follow-up on what I showed before, This is intake we had. I showed this slide before. That was how it looks when you come to the end of the small intestine. And this is, on average, what it looks when you come to the feces. And this is actually how it looks when you try to plot them. This is the ileal data, and this is the fecal data. You'll see that they have the capacity to metabolize this form of nutrients. The next part, which I'll move over to, that is what is happening when we change the diet from a concentrated diet to a more fibrous-rich diet in terms of nutrients of carbohydrates, associated nutrients that is assimilated. Well, this is from studies that was performed in the Netherlands, and there is also a couple of our own studies, and more is actually around in the literature. But these two I've chosen this set of data here because it very well illustrates what's happened when we are going from a situation where we are switching from a concentrated starch-based diet to a diet containing more fibrous component. And what you can see here, if you are looking on the proportion of energy, of carbohydrate energy that comes from glucose, you'll see as more of the fibers are in form of non-digestible carbohydrates, you'll see a decline in the uptake or energy provided to the animals as glucose, and simultaneously increase in short-chain fatty acids. And this is basically what we see across different studies. Most of the diets which we are dealing with, practically they will lay down in this area. So you can see this is really, this is just to go to some extent to the extreme. What's happening is that, this is the carbohydrates that can be taken up by or can be broken down by the industrial enzymes. Here you see them coming up as glucose, fructose, and galactose, in most cases glucose, whereas those which are fermented will be absorbed as the short-chain fatty acids. Trying to calculate on what does that mean? Well, first of all, if you're looking into equations for the fermentation of carbohydrate into short-chain fatty acids, you will see that you lose in the order of 25% of the energy. That is going up because they are present as some of the fermentation gases. Another thing is that if you're looking on how efficient the different short-chain fatty acids are metabolized relative to glucose, for one or another reason, there is some data up here that's disappeared, but this is efficiency relative to the efficiency of glucose. You'll see that the efficiency for the short-chain fatty acids in the intermediate metabolism, that is lower than of glucose. So whenever we pour more of the digestion towards the lower gut, we'll not only lose energy because it's more difficult to degrade, but they will also generally have a lower there will be a lower utilization of the energy. And if I try, this is data which is not published yet, but if I try based on carbohydrate or fibrous composition of feedstuff from our feed database, try that or to relate that to both digestible energy and also metabolizable energy, you will see a decline in the in metabolizable energy as more of the you have more of the fibers in the diet. This is primarily driven by the digestibility parameter, less digestibility of these diets with high levels of fibers. Another aspect which is also and which relates to what I was talking about before, that is if we try to plot metabolizable energy, or net energy as a percentage of metabolizable energy, then we will also see that we have a decline in utilization, because as more fibers or the feed has more fibers, more of the nutrient will derive from microbial fermentation in the large intestine, more short-chain fatty acids, lower utilization than is the case with glucose. I have some time now. Yeah. This is just to try to wrap up what I've been talking about here today. And I've tried to put a little bit into grain perspectives. If we look, and why a grain? Well, first of all, if we look on feeds for pigs, grain, that's the major part of it. And I have just tried to illustrate it a little bit with how it will happen when we have a grain that passes along the large intestine. We have this is just wheat, and you can see some of the cell structures up here. We have the protein, fat, starch, and also the fibers components. They pass along the length of the small intestine. What disappears, that is of carbohydrate-derived nutrients. That is degradation of starch going into the body as glucose, first and foremost. What we find down here in ileal digester, that is a collection of fibrous components, soluble and insoluble, and also some encapsulated nutrients. From a cereal grains, it will primarily be nutrients that's present in particularly the aileron part of the grain. When this digester passes further down through the large intestine, all the soluble components, they will be gone. A good part of the fibrous components that's present in non-lignified materials, that means, for instance, the fibers that's present in the endosperm, most of the fibers that's present in the aileron cells, they will be gone during that transit through the large intestine. Whereas the outer part of the grain, most of what is present in the outermost part of the grain as cellulose, in particular lignin, and some types of cell wall bound protein, they will be excreted in the feces. I should also say here, along with the transformation and the products that's absorbed, that is as short-chain fatty acids. I should also say that along with that process, there will also be some microbial protein that's excreted. What is happening if, for instance, we take a concentrated grain and then add to that or substitute part of the grain with a DDGS? As you may remember from the presentation of Dr. Stein, the DDGS has a relatively high content of fiber. What is happening to the diet? Well, the starch goes down, fiber goes up, amount that pass to the end of the small intestine goes up, amounts that pass all the way down through the intestinal tract goes up. But there will also be part of it that will be and the consequences of these changes, that is uptake of glucose goes down, more of the carbohydrate assimilation products as short-chain fatty acids, but at the same time there will also be a reduction in metabolizable energy or lower, and the same is the case with net energy. That is briefly my presentation in a nutshell. Well, first of all, at last I would like to acknowledge my funding bodies. A lot of what we have been doing has been funded by the Agriculture and Veterinary Research Council, some industries, and then I also want to acknowledge my colleagues at the Institute and also my technical staff. So thank you very much for your attention. This is where I come from.